My name is Ahmed Elkady. I'll be presenting a study that has recently published in the Journal of Magnetic Resonance Imaging entitled Longitudinal Multiparametric Quantitative MRI Evaluation of Acute and Chronic Multiple Sclerosis Paramagnetic Limb Lesions. I have authored this study with a group of colleagues from Montreal Neurological Institute Hospital, New York's Research, Roche, Genentech, and McGill University. Multiple sclerosis paramagnetic limb lesions, or MSPRLs for short, are important MRI-derived markers of chronic active. Histological evidence supports that PRLs exhibit limbs with iron-loaded macrophages and macroglia and cores with prominent demyelinating features. Multiparametric studies of PRLs and the interdependence between core and limb are lacking. The purpose of the study was to apply whole lesion detailed multiparametric analysis to investigate cross-sectional characteristics of acute and chronic PRL and non-PRL lesion types, evaluate the longitudinal evaluation of acute and chronic PRL and non-PRL lesions, and finally, study the correlation between core and limb of acute and chronic PRLs. Our study design consists of 95 progressive MS subjects with at least one persistent PRL. These subjects were retrospectively selected from CONSONANCE clinical trial studying the effectiveness of occluzumab in progressive MS. The subjects were imaged at screening, week 24, week 48, and week 96. Acquired a number of MRI images at 3 Tesla. So you see an A T1-weighted image acquired prior to administration of gadolinium contrast, and B, you see triple spin echo T2-weighted image. Both A and B were used to construct C, which is the standardized T1 over T2 weighted image. D and E show magnitude echoes 1 and 6 from the gradient called echo acquisition, which were all echoes were used to construct F, which is the star map. And also G and H show the phase echoes from the gradient called echo acquisition, and all phase echoes were used to construct I, which is the condensability map. The slides demonstrates how we classified MS lesions. You can see in the first row, screening acquisitions were flared post-administration T1-weighted post-contrast administration T1-weighted images and the frequency image. And the second row shows overlaid manually drawn PRL limbs in green. The third row shows the same images, but for week 96, and the fourth row shows the overlay of the manually drawn PRL limbs. So you can see that the dark blue arrows are highlighting a persistent PRL lesion showing PRL limbs at both screening and week 96. But they also displayed enhancement as a hyperintensity in T1-weighted post-administration of gadolinium contrast at screening. The light blue, on the other hand, is highlighting a lesion that showed enhancement at screening in the T1 post-administration of the gadolinium contrast, but did not show limbs at both screening and week 96. The yellow lesion shows a persistent PRL that showed limbs at both screening and week 96, but did not show enhanced with a T1-weighted image at screening. Finally, the orange arrow is pointing to a lesion that did not show a limb at both screening week 96, which thus was classified as non-PRL or non-persistent PRL, and also did not display enhancement at screening in the T1-weighted post-GAT image. I will be highlighting in the next slide as a zoom in of the dark blue lesions that were highlighted by dark blue and light blue arrows. So you see the zoom in preview of the gadolinium persistent PRLs and gadolinium non-PRL for FLAIR post-GAD T1 weighted standardized T1 over T2 weight ratio images, ultra-star frequency, and quantizability. You see also overlaid are the persistent PRL GIMs, which were drawn or computed using intersection of the screening recognizer manually drawn GIMs shown in the previous slide, and cores were drawn or computed using a filling of the manually drawn PRL GIMs, which were intersected with automatically segmented T2 lesions, and intersection of both screening week 96 core labels were used to compute the third row, which is the persistent PRL core labels. And finally, the persistent whole lesion labels were computed as the union of both GIM and core labels. In terms of statistical analysis, we used an encrypted threshold of alpha equals 0.05. And we employed repeated linear mixed models to study fixed effects of time and lesion type using two types of models: discrete changes to study discrete changes using clear model, and the second type of model we used was a continuous model to study linear trends between time points. We employed two labels, one whole lesion labels, and two the detailed PRL labels using separate GIM and core labels. Finally, we did the correlation analysis between GIM versus core to study the relationship between GIM and core lesions. So now preview some of our results. So you see in the first row, you see the results of the acute lesions, which are newer lesions, and the second row shows results of chronic lesions, so are older lesions. You can see that both the acute and chronic lesions showed lower ST1 over T2 weighted ratio values and ultra-star values for PRLs compared to non-PRLs for both acute and chronic lesions. This indicates that the PRL subtype is more demyelinated than non-PRL subtype. You can also see that there was positive QS values in chronic PRLs compared to negative values in non-PRLs, indicating the higher iron content for PRLs. You can also see that those acute PRLs had steeper positive slopes at screening 24 period for all the three parameters for acute lesions, and this indicates that there's an improved partial recovery of acute PRLs compared to acute non-PRLs. In terms of detailed analysis of PRLs, you can see that core and GIM labels produced significant results or significant trends that were not detected using whole PRL labels. And this, for example, you can see this prominently in ultra-star analysis in this slide. Also, we found significant correlation between GIM and core using all three parameters we employed, which indicates an interdependence between GIM and core PRL biology. In conclusion, longitudinal evolution of PRL subtypes using standardized T1 weighted or T2 weighted ultra-star and quantitativity may provide valuable information about chronic active biology and progressive MS. Additionally, detailed PRL analysis provides valuable information not provided by whole lesion analysis. We'd like to acknowledge funding from Roche, United States Department of Defense, Canada First Research Excellence Fund, and MITEX Accelerated Program. And finally, we'd like to acknowledge and thank all patients, their families, and the investigators who participated in the CONSONANCE clinical trial of tislelizumab.